Good evening. For those of you who don't know me, and I guess actually for those who do know me as well, I am Boston University Provost David Campbell, and it is my distinct pleasure to welcome you all here tonight to our annual University Lecture. The University Lecture Series began in 1950, and over the past 59 years has offered members of both our Boston University community and the general public the opportunity to hear from many of our most distinguished scholars. This forum is meant to honor members of the faculty who are engaged in outstanding research and scholarship. Each spring, we invite members of our faculty to submit nominations for the annual lecture, and those recommendations are reviewed by a nominating committee made up of the university lectures from the 5 previous years. But the university lecturer does more than merely honor a single outstanding faculty member. It provides all of us with a chance to celebrate the quality of our faculty by hearing directly from one of our highly accomplished scholars who is engaged in work on a topic or issue of great importance. As you know, some of you perhaps all too well, every day hundreds of lectures take place at Boston University in classrooms and halls throughout our campus, delivered by members of our faculty, visiting scholars, and public figures. Tonight's university lecture, however, is a singular event, not only because of the rigorous process of selecting the speaker, but also because of the rich history behind the series and the work spent both in preparing the lecture and in documenting it. The university lecturer is, in every way, a celebration of Boston University's intellectual life. We are both proud and delighted tonight to present as our 2009 university lecturer Professor Thomas Kunz, biology professor, director of the Center of Ecology and Conservation Biology at Boston University, and a faculty member since 1971. Professor Kunz's research focuses on the ecology, behavior, evolution, and conservation biology of bats. What follows is a partial listing of his distinguished accomplishments in this field. He is the author or co-author of over 240 peer-reviewed publications and is the editor of Ecology of Bats and Ecological and Behavioral Methods for Study of Bats, and co-editor of Bat Biology and Conservation, Bat Ecology, Functional and Evolutionary Ecology of Bats, and the second edition of Ecological and Behavioral Methods for the Study of Bats. He is an elected Fellow of the American Association for the Advancement of Science, past president of the American Society of Mammalogists, a recipient of the Garrett S. Miller Jr. Award for Outstanding Research on Bats, and the C. Hart Merriam Award for Outstanding Research in Mammalogy. He was the 2008 recipient of a Lifetime Achievement Award from the Karst Waters Institute and was elected to honorary membership in the American Society of Mammalogists, the highest award given by that society. Most of his past and present research has been funded by grants from the National Science Foundation. His current work focuses on the analysis of collective behavior of bats, assessing the ecological and economic impact of Brazilian free-tailed bats on agro-ecosystems, assessing the impacts of wind energy development on bats, and assessing the effects of the white nose syndrome on hibernating bats. This disease has led to a most precipitous decline of bat populations in recorded history. He has conducted field research in North America, Costa Rica, Ecuador, India, Malaysia, and the West Indies. He is the founding director of the Tipituni Biodiversity Station in eastern Ecuador. He has promised tonight not to release a bat into the audience. He described the last time that he did it, that the bat flew around, closely following the path of anyone who raised his or her hand, and then landed on Tom himself in a very delicate place. I will leave that for your... It is now my pleasure to present Professor Tom Kunz and the Aeroecology, the New Frontier. Tom.
Thank you, David, and thank all of you for coming. I should also thank David for giving half of my slides here. So now I can, or maybe all of them. Are there any questions? I'm going to go back to the beginning. Okay, sorry about that. I think the keyboard here was a little bit more sensitive than I think David thought. In any event, it's a great pleasure to be here, and I'm very honored by this award. And again, I can't take credit for all of it. I must give credit to former students and current students, postdocs, and colleagues who have really assisted in many of the things that we've done. So aeroecology is really a new term that I coined 2 years ago based on a conference symposium I organized. It was held in San Antonio. And it grew out of my interest. I actually have a slide out of place. Let me make further acknowledgments here. I got thrown off here by these other slides. Anyway, going back to state how many people have really helped in this. And this is a partial listing of current and relatively recent students and postdocs in my lab, as well as recent research collaborators. And it's none of this could have been done without the work and help of these folks. So in many ways, many new disciplines have evolved and developed over the last, centuries, actually. And new disciplines arise largely as a consequence of these four areas here of intellectual pursuit. And the metaphor of the alignment of the planets is really what makes it possible for new disciplines to emerge. And those are interest, technology, empirical research, and theory. And I believe in many ways, many of us, apologies to my former students who were actually doing aeroecology before they knew what it was. It's only recently we really just put it into perspective. What really drove me to come up with this idea was in 2004, I was asked to serve on a National Research Council committee to evaluate the impact of wind energy development on birds and bats, because there had been a number of large numbers of these critters killed, particularly in the Appalachian ridgetops in West Virginia, Pennsylvania. And that 2-year experience was very valuable to me, but what I did learn, and as well as my colleagues on this committee learned, that we really didn't know very much about what occurs above our feet, that is, into the airspace above us. And part of the reason we didn't know very much about this is because we didn't have the tools or the technology to do this. And it's a great challenge to actually do some of these things. Now, I'm going to this composite illustration here basically shows there are 3 species of bats on the left that are the most frequently killed. These are migratory tree bats. I'm going to go back a minute because there are 2 little video snippets here that are going to be difficult for you to see. I want to play them. The one on the upper right is a stationary turbine. And these are big turbines now. These things are as high as a football field is long. So they're very big turbines, and so a little bat is dwarfed, the size of my thumb is dwarfed, but you can see one. And we used a thermal imaging camera to actually see, and the first one shows a bat sort of curiously flying around this happened to be a turbine that was not functional at the time, and then the other one below you'll see a direct hit. It turns out most bats are not killed by direct hits, but killed by what is called barotrauma. That is, the bats get caught up in the turbulence around the blades. So watch, if you can see the first one here. Let me go back a second, and now we'll start it again. And see if you can see a little bat. Can you see that little bat flying behind the pole, comes out from behind it, and then flies around the stationary turbine. The next one you see a bat gets whacked there. And my apology is we can't get any closer than this to these with the thermal cameras. In fact, we need 3 cameras to cover the entire field of view of these. Well, so what is aeroecology? Again, it's a study of interactions of organisms in and with the aerosphere. And I'll define the aerosphere here in just a second. But it includes the study of passive aerial movements of viruses, bacteria, spores, pollen, seeds, and some spiders. It also includes the study of active movements of pathogens and pollen and spores and seeds by insects and birds and bats that use this environment. It includes the study of migrations of insects and bats and birds in the context of the atmospheric conditions that prevail at the time. It also includes aerial feeding ecology, that is, of animals that feed on other animals, such as frogs in this case, or those that feed on fruit, as in this particular case here, or those that feed on fruit like eating corn on the cob, and here's a nectar feeding. And then there are also birds that feed on bats. This happens to be a red-tailed hawk feeding on bats that we've studied at a site in Texas, and this happens every night. So there's a lot of activity going on in the sky or in the aerosphere, and that's something that we need to, we haven't really had the technology in many ways, particularly at night, to do this. So what is the aerosphere? It's the boundary layer of the Earth's atmosphere that supports life. Life actually doesn't exist way, way up there in the stratosphere, nor does even the upper atmosphere, even in the upper troposphere. It's the lower boundary, very lower part of the boundary layer. It's the least understood part of the biosphere that interacts with the lithosphere, the land, and the hydrosphere, water. It includes movements and unique characteristics of gases, water, and dust. It includes active and passive movements of viruses, bacteria, spores, pollen, seeds, and the feeding interactions between arthropods, birds, and bats. The aerosphere is a truly circumglobal environment in contrast to the Earth's land masses and oceans that are broken up by one another. It is a highly variable dynamic fluid. It experiences enormous geographic, daily, and seasonal variation in weather, and altitude, celestial forces, day length, geomagnetic and gravitational forces. And it's these abiotic selective forces in the aerosphere that have strongly influenced the evolution of specialized forms and function in organisms that depend upon this fluid environment. Form, for example, shapes. If you think about the shape of birds and the sort of fusiform shape that minimizes drag, and the size of their bodies, the shape of their wings, the specialized organs, the various sensory systems and respiratory and vascular systems are adapted to this particular environment. The little illustration in the far right, you might be able to see, which happens to be an illustration of a bat wing, have little sensors, just like you have hair on your hand, and associated with each hair is a little muscle and a little sensory tactile sense. Those bats, as they fly through the air, sense the air flow around those wings that make it possible for them to navigate as they do. There's also function in terms of thermoregulation and water balance that is unique in many ways to the birds and bats and insects that use the aerosphere, and various kinds of feeding and navigational cues that land-based animals and aquatic-based animals really don't use. And reproduction in terms of litter size and clutch litter size in mammals and insects, and clutch size of birds and so on. So the evolution of powered flight has made it possible for most insects and birds and all bats to exploit the aerosphere. And here we're seeing an evolution of 55 or 52 million years of evolution of powered flight condensed into 15 seconds. And this is something that we don't know a lot about. This was we did a program with National Geographic a few years ago, and this was some creative artistry that was done as part of that video. So why should we care about the aeroecology? Well, many of us are interested in climate change, air pollution, altered landscapes, altered aquascapes, declining insect populations, declining bird populations, declining bat populations, emerging pathogens, invasive species. What's next? What about nanoparticles? What's going to happen when these nanoparticles are released in the environment? We don't know. We didn't know when we released chemicals 40 years ago in the environment. So these are sort of heads up on what can happen and what we need to be concerned about. So aeroecology as a discipline is a paradigm for cross-disciplinary research. And I was trying to think of the kinds of people that I've interacted with over the last 10 years focused on many of these kinds of questions. And much of my research today involves people, atmospheric scientists, other biologists, computer engineers, computer scientists, earth scientists, electrical engineers, geographers, mathematical modelers, public health scientists, statisticians, social scientists, wildlife managers. So it's really a composite, a cosmopolitan group of people to answer questions that most of us haven't really thought too much about. And the major advances in this discipline are being conceptually and technologically driven, as many other disciplines in science. That is, and just to list a few here, remote sensing technologies, radar imaging, digital photography and high-definition videography, night vision and thermal infrared imaging, radio telemetry from both aircraft, satellites, and cell towers, and acoustic detectors, both audible and ultrasonic, use of stable isotopes, use DNA and hormone assays. These are all technologies that my lab has used in various ways and continues to use. So the questions and challenges for aeroecologists, and if you haven't become one yet, hopefully you will if you have that interest in whether it's in abiotic or biotic aspects of the aerosphere, is how best to detect the presence, diversity, and activity of organisms in the aerosphere, how best to quantify these variables at different temporal and spatial scales, and how best to understand and interpret the responses of organisms in the aerosphere under different meteorological and atmospheric conditions and anthropogenic perturbations. Jumped ahead there too fast. Okay, so let's hit there. Okay, there are many things that if you look around our environment, there are challenges to the organisms that use the aerosphere. Those that disperse, feed, migrate through an ever-changing aerosphere. Here's a bird's eye view, for example, of the aerosphere, of volcanic eruptions in Montserrat a few years ago, dust storms over western Iraq or other parts of the world. This is what's happening and certainly have happened for millennia, but nonetheless, many of these things are happening more and more. This happens to be one. This is the bat's eye view of a lighted terrestrial landscape from the aerosphere. Most organisms have evolved their physiology, their reproduction, their timing of migration to light cues, and how does our polluted, lighted environment influence these, and how will it affect them as years go by? Interactions as you're flying through, organisms fly through the aerosphere, they obviously, whether they're diurnal or nocturnal, see different things. And they may, many times, migrating birds and bats stop over at places for short periods of time to refuel. And many of the landscapes that they were originally used to are no longer there, or have been grossly altered. In the case of surface mining, irrigated croplands in Colorado where waters are drawn from aquifers and dumping salt on the surface and so on. And tar sand mining in Canada, bats have been found stuck in the tar in some of these areas because of thinking that looks like water to them. It's a nice smooth surface in places. Rice paddies in India, in other parts of the world. Interactions with urban landscapes and aquascapes. It's no surprise that birds and bats are killed by high-rise buildings throughout the world and collide with bridges and other structures in the environment. Also the interactions with air pollution and in the aerosphere. And smog, in this case, in upstate New York, attributed to the combustion of fossil fuels in the Midwest. Aerosols over the Pacific Ocean, attributed to industrialization in China. What happens in China 4 days ago ends up in San Francisco a short time later. The increasing threats to airborne fauna in the aerosphere from commercial and military aircraft. The Smithsonian logs what species and what critters are hit by aircraft. And surprisingly, as air traffic increases, more and more of these little critters are being killed. Tall glass buildings seem to be an attractant to many birds and bats as they fly, as well as insects. The building on the right is interesting from Malaysia. Here is a new high-rise building which also has wind turbines on it. And so it's a double jeopardy in that case. So the worldwide proliferation of other structures that extend into the aerosphere, including communication towers, utility-scale wind energy facilities, these are, in fact, altering what organisms that use that environment. In fact, wind turbines, as I pointed out earlier, wind turbines are responsible for killing unprecedented numbers of migratory bats and birds. Look at the field of wind turbines here. Now, I'm the first one to say I am not in favor of keeping our current carbon footprint. We need to reduce it. We need to find ways to do this. But we just simply can't go out and put up develop a new technology and put it up without, first of all, finding that there is some impact of this on wildlife, including birds and bats, but other critters as well. So traditional and novel technologies are needed to investigate aeroecology of insects, birds, and bats, as well as other creatures like spores and seeds and microbes and so on, to better understand the ecological and economic importance of mankind. Tonight I'm going to increasingly focus on bats because that's my focus, but I wanted to try to give a little broader perspective at the beginning here, because one of the services, one of the values that bats provide is they suppress insect pests. They do feed on insects. They're also species that pollinate flowers. Those of you who know something about the century plant or the agave, the distillate of that is something you probably partake of periodically if you've ever had a margarita, that is the tequila that comes from that distillate. And their contributions to transmission of emerging diseases. We know that birds and bats and insects do carry some of these, or transmit some of these pathogens to humans, for example. So collaborations of biologists with atmospheric scientists, computer scientists, et cetera, as we've talked about before, we need to, these collaborations are essential if we're going to meet important challenges that face us all. So probing and modeling the aerosphere for airborne fauna, again, advances are made using some traditional methods and novel recordings and analyses. I'm just going to look at a few. Mist nets, for example, are one of the common tools that people who study birds and bats use to capture them, often at ground level. And we can learn something about the behavior of animals that feed that close to, or fly or drink that close to the ground. This was a little slide that based on some work I did way back in the early 70s. Discoveries of nocturnal activity. When are bats active? Are they only active right at dusk, or are they active at other times of year? This has important implications for understanding a lot of things about the behavior and the ecology of these bats. This particular slide, new discoveries are being made with the deployment of canopy nets. Canopy nets, for example, we can get as high as the ceiling here in this building, and we can capture critters that we wouldn't otherwise be able to do just putting them on the ground. And this slide shows you that a study we did in Peninsular Malaysia and published in 2003, over 60% of the bats and species were captured well above the level of ground nets. And one particular species which was only captured in the area that I've shown here in green are a species which we were the first to describe, a wild mammal in which males lactate. And so discoveries like this can also add an understanding about the physiology of humans, for that matter, of understanding why don't males lactate. Many of the women sitting in this room probably would like for their husbands to have lactate. They just wouldn't like the clothing they'd have to wear associated with it. But anyway, their novel advances in ecological and behavioral physiological research derive from captures of another technology. It's a very simple technology called a harp trap. This trap looks like a harp. It has a bag that hangs beneath, partially lined with plastic. And to take a bat out of a mist net, you have to kind of pick it out like this. Here, you just simply take them out of the trap with your hands, and the time element is important here because many kinds of studies, particularly using hormones and other assaying hormones, such as stress hormones, glucocorticoids and so on, you need to sample and take the blood within 3 minutes of capture. It's possible to do that with a harp trap, not with a mist net. Using various kinds of ultrasonic detectors that extend beyond the ability to capture with a harp trap or a net has made it possible to identify species of presence in certain regions that we wouldn't have otherwise been possible to do. And it's also using these in combination, using a combination of 4. If we had microphones placed around this room, my voice travels at different rates depending upon where I am relative to the microphone. And so some biologists are beginning to track an animal in space based on the time frame which the sound is coming to those microphones. Using what are called PIT tags, or passive integrated transponders. These are little, about the size of a grain of rice, often used in laboratories to keep track of mice and rats and so on, or also used in fish in rivers and so on. These little tags are injected under the skin. This little loop antenna that's shown in a corner of a barn in New Hampshire, I don't know if Gina's here, but maybe I'll put one on one of her barns, and we'll try to see if she's got bats there, and if they do, we can keep track of them. The illustration on the right, the bluish one, shows one seasonal record of the sort of emergence patterns. And what we were asking whether in this particular study was whether babies followed their mothers on their first foraging bouts, among other questions. But that's something you can do with the little tags that keep a record. They're unique alphanumeric numbers of those tags that make it possible to know what individuals are doing. One of the grand challenges that are faced by not only aeroecologists but all ecologists is how many of anything there is. It's also a question that many of us as administrators and so on ask about how many students are coming to the university next year. This censusing is important for studying wild animals as it is studying humans in any context. And there are many ways we can do this. Sometimes it's very simple. We can count the number of students coming out the front door of the Psi Auditorium, how many came tonight, or how many students passed across a certain area. But we can also do that with small numbers of bats. We can do a direct count. In some cases, we have to make an estimate of the count. For example, in the lower right, we simply take quadrats and measure how many individuals are in a given quadrant. There are limitations to this because the distribution of the animals across in that particular space is not uniform, as you can see here. So there are biases associated with that. Censusing bats and getting a little focused more on what I've been doing here in the last number of years is, again, censusing Brazilian freetail bats. These bats form some of the largest aggregations of mammals known anywhere in the world. Any of you have been to Austin, Texas? The Congress Avenue Bridge has probably close to 1 million bats living under it. These bats play an important role in agriculture in terms of suppressing insect populations. But the challenges of counting them are enormous. First of all, in the roost, if you go into the roost, you can't, the walls are uneven, you could disturb the bats, and that's not good either. They roost at densities of several hundred per square meter. The nightly emergence rates, as shown here in the middle slide, shows what emergence might look like in some of these sites, whether it's the Congress Avenue Bridge or caves, averaging thousands per minute passing in front of your eyes. And the problem is these bats sometimes emerge early in the evening, but they continue on until several hours after darkness. So the next view, if you had no technology other than your eyes, that's what you would see. You'd see a black box. And so that's one of the challenges that we faced. We've studied Carlsbad Caverns. Some of you have been there before. Let me see the hands that have been to Carlsbad Caverns. Anybody? There are two big things there. There's the caves and the cave and the bats. What you just saw there was the emergence, the pre-emergence behavior of bats. They go through this spiral before they emerge. And this is sort of standing inside looking out of the cave here. But I'm going to ask you to count these for me visually and tell me how many you think there are. This happens to be a study site of ours that actually the Nature Conservancy took over a few years ago because we were tired of students or people coming out there with their beer cans after 3 or 4 beers asking how they can help us. And so we decided and we convinced the Nature Conservancy to build a grandstand over them to get them out of our way. Anyway, how many bats were there? Anybody get a count? Okay, well, there are about actually 6,000 there. That was a 20-minute snapshot. Okay, I'm going to tell you how I know that in just a minute. And one of the ways we've met this grand challenging of censusing, whether it's day or night, is to use thermal infrared imaging. And this just shows the infrared spectrum. We use the mid-range, the cameras that we develop. The upper right is a portrait of yours truly. And some people have told me, well, as you can see, this is in false color. Thermal imaging doesn't see through glass or plastic, but the colors, the false color here shows, again, blue. The blue sky is the coldest. The clothing, I'm insulated. My hair insulates. My forehead is red, and my nose is green, which is cooler, the coolest colors. So I have a cold nose, sort of mid-warm cheeks, and a redneck. And some people call me a redneck because of that. And that's just because I've got nice, hot vascularity there. Well, the sensitivity of these cameras is actually pretty amazing. As you can see, the temperature sensitivity of the highest end cameras, which we just happened to get 3 of them recently from a grant from the Air Force, that records temperatures about 200 degrees Celsius. And that's really quite amazing when you think about these cameras, at least the newer versions. So this is the way we census bats. It sounds like a very simple thing to do, and it is, but it's challenging in many ways because every place is different. Where we set up cameras, for example, we try to set the camera perpendicular to the field, the flow of the bats. We take our camera, the computer sits out in the field. We have to haul all this stuff to the field and make sure we have generator power and so on in remote places. So this is a raw thermal image of emerging bats at the beginning of an emergence. And you can sort of see little pink bats. You might not see them very well. But working with colleagues in the computer science department, in particular Margaret Bitka here and her students who have helped develop algorithms to detect the presence of these thermal hotspots, that is the warm pixels in the digital image, and then also track these bats. And by doing this, it's possible to then actually digitally count them as well. So the next slide basically shows some of the algorithms, the graphics of the algorithm, so we can actually see, for example, in the upper left, you see a peak of a single bat. Over on the right, upper right, you see multiple bats. The hot spots are yellow, as shown in the illustration on the left, upper left there, the warm body and the cooler wings. And then you can discriminate this, and the computer algorithms in an area of computer science called computer vision is possible to filter out stuff that we really don't need, such as vegetation. Okay, so these are all those bats you saw before, but now a detecting algorithm is applied to each, and so we can see them much more readily. The reason we couldn't see them all equally before is because there's a depth of field of view. The bats that are closest to us have more pixels than the ones farthest away, and so they're going to appear hotter than the ones farther away. But this algorithm makes it possible to actually distinguish those. And is Diane Hirsch here? Hi, Diane. Diane was an undergrad here, working with Margaret on a project where you took her to the field. Not many computer scientists go to the field. Diane did, and she helped us develop the next algorithm here, which was the tracking algorithm. And so basically what we do is add little tails to these little marked bats, and then over to the right, as they pass through the field of view, they're counted. And appropriately so, she called these, of her generation, called these Fruit Loops. I never ate a Fruit Loop. I used to eat Cheerios, but Fruit Loops are colored Cheerios, and so this is how they came about being called Fruit Loops. We can, in larger concentrations, these are actually detected bats in a much larger emergence. As the night progresses, the more and more bats come out, and then if you add the tracking algorithm to these, we can begin to start seeing things that are really quite interesting, and we begin to understand the dynamics of this group behavior or collective behavior, as David mentioned earlier. If you look at that, initially, I'm going to go back a second to this. If you look at this, we all know the Bernoulli Effect, and we postulate that the bats in the middle of this column are actually moving faster than those on the edge because of the friction, the air friction, and the ones in the middle maybe have an advantage in that they take it, just like migrating geese and so on, take advantage of the airflow of the critters in front of them. But if you just take a quick look at that, you sort of think it looks like there's a lot of activity in the middle unless it's slower on the edge. And I won't give away the story right now, but there's the guys and gals in the computer science department are helping us try to answer those kind of questions. We can actually partition out individual bats to understand what's going on, whether they move in a straight line or they move. And this is just important in terms of understanding just what is the behavior of these animals in these groups. Why do they form these columns like this? Why do they just not go out every which way? One hypothesis is it is a predator-deterrent tactic. When it's light out in the evening, red-tailed hawks are less likely to dive directly into a deep column, dense column like this. So understanding how individual bats move is very important. This is the most recent analysis of 3D tracking. All that previously was 2D tracking. This was Zheng Wu and others in the computer science, Margaret and some other colleagues here have developed a way of tracking bats using three thermal cameras trained on a certain area. And so each color of the bat is, in each of those frames, are tracked. And in the lower right frame, you can see what the trajectories are. This is the first and the very beginning of trying to understand group behavior. And we hope to expand upon this now that we have these new cameras and in the near future. Well, we can also count bats, as I said before. We count bats. This happens to be 3 profiles of the Emergence Peaks at Carlsbad Caverns, which we have worked on for 3 or so years. But as you can see, there's high variability within just a few days in April of 2007, not only in terms of numbers, April 11th of 63,000 bats, and April 12th, 123,000. Bats move through these. We learned something about migratory behavior. April 17th, it dropped down to 54. So we assume that these bats are probably moving through and stopping over at Carlsbad for a period of time. I want to take you to Carlsbad Caverns for a minute because there's those of you who have been there or haven't been there, you should go. It's based in southeast New Mexico. The face of the cave looks like this. On any one night in the summer, you might have 2,000 people sitting in a grandstand where they're looking directly into the cave, waiting for the bats to come out and waiting to see what they do. And one of the things that we have been asked to do, and we were asked to do, is really validate and whether bats are declining there or whether they're increasing, just what it is. Well, as we look at the literature, the colony size estimates are all over the place, ranging from 3 million to 8 million, 8.7 million, down to a few thousands here. And again, most of these are based on a single night estimate, in some cases a few nights, never over the entire season. And so you're comparing apples and oranges when you don't really have a consistent way of doing this. And so one of the things they paid for us to do this, and we were there 3 years doing these. And this is 1 year. We have 3 years of data like this. And these are the Emergence Peaks. And in the far upper a line right here basically shows the seasonal changes. This is that flux that came through in April, and then that's pretty much stable during the summer. This is a very strange drop that occurred right here. This was a very dry year. For some reason, most of the bats left the cave for 1 week. Where they went, we have no idea. They came back, and then there's a continue on throughout the season. Now, one of the things about Carlsbad Cavern, in addition to the bats, it's an amazing place. It's something, this is a laser-generated model of this cave. Something like, I can't remember the exact number of miles, 54 miles of chambers here, underground, nearly 1,000 feet deep. The yellow part that you see here is actually what is called Bat Cave. This is where these freetail bats live. Looks like Star Trek, doesn't it, sort of? Okay, so here is this yellow area where the bats are coming out. This is a model. This is actually what you're seeing right now. Those little red dots are a model. And we use the model to test the hypothesis that these early estimates of 8.7 million bats was bogus. That was our hypothesis, because we can't imagine all of our estimates that we did with thermal cameras at this site, on average was anywhere between 500 to 600,000 bats on a given night. And the guy who, his name is Allison, described how he estimated this 8.7 million. The only paper out of the 6 or 7 that were there that actually very explicitly said, this is how long the emergence was. This is my assumption about how much space a bat takes up. So we modeled this. Okay, we also modeled our own data. And we used a model called Maya. Some of you may know it. It's an algorithm that was developed basically for Hollywood, for animations, and it was, in my understanding, it was developed at MIT. Bob may know that. So what we did, actually, to extend that particular model of the cave, we actually took laser measurements ourselves. That whole cave was laser mapped, not by us, but by the natural resource people at Carlsbad Caverns. But we took a laser we took laser measurements and recreated the entrance of this cave, because we needed to know the size and shape of it to actually do a better job of finding out how the bats behave there. And this is the model of, again, it shows the bats, how they would come out. And one of the important things to note in this is that there's an eye of the needle right here. They have to be forced through that eye of the needle to come out of a much bigger opening. Okay, so what we did was, first of all, took our own data, and this is that same opening. And what we did, we simulated the emergence flow rate of 18,000 bats per minute. And that's about, on average, of what we have been discovering over the few years. And this is what these modeled little bats, they take up the equivalent of 1 cubic foot. Okay, and they go through that spiral. Remember we saw before, they're going through this, from our perspective here, the counterclockwise spiral. From their perspective, it's a clockwise spiral, and they go out. And so what we do is census the bats as they're moving through the field, out of the field of view here. And this is typically what we see. This was a night of 42 minutes that yielded about 756,000 bats on that particular night. Okay. this is what, in using the data that Allison provided for us, this was his flow rate was 1,000 bats per minute in a 23-minute period that actually would have yielded closer to 11 million bats, and here they come. Okay, this is, now it's very unlikely that bats could possibly even fly that close to one another as they're all mushed together, and they don't never, ever have seen them fly out of a cave like this mass seen here. So to further test this, and we wanted to compare his estimate with what our estimate, and there's our estimates data. His estimate is shown in the red line. In order to get his data onto our profile of emergences and so on, we had to reduce ours down to almost a pinhead to get his data into the so this is again a test of a hypothesis that I think we've demonstrated pretty clearly that his estimate was bogus, and there wasn't the massive decline in that particular species as was predicted. Now, testing a conceptual model is something I've been interested in for a long time, and that is what we call refuging species. We are refuging species. In many ways, we refuge during the day at BU, but we spread out and disperse at night to go to bed. Bats do the different. They sleep in the day during the caves, and they disperse out at night to go feed. In some ways, very similar to what we do, but they feed. This particular model, again, showing the cave is the refuge, and there are corridors or trampling zones, like the gray area would be around the city, and the corridors would be the highway is going out. And the habitats shown around here, different kinds of habitats in south central Texas, live oak, mixed forest and agriculture, and then strictly agriculture. Simply in this cartoon illustrates this. One of the things we wanted to do is test some particular predictions about this, about a refugee species. And that is, we would make the prediction that foraging times for individuals in a large colony should be highly variable. Just as it is, some of us live far away, some live very close, and that's not it's a kind of a no-brainer sort of prediction, but nonetheless we wanted to find out is this in fact the way it works. Our foraging distances and directions for individuals in a large colony should be highly variable. Daily energy expenditure for these individuals obviously should reflect this variability. And so one of the things we've taken advantage of is a Doppler radar. Now, you've all seen Doppler radar in your weather channels or whatever newscasts you watch at night. And interesting, in this area in New Braunfels, Texas, and there are 144 of these across the United States, but this happens to be near some of the caves. And what you see on the right is a series of, at 15-minute intervals, what on a clear night the radar technicians would have seen. And one time they called us and said, we went outside and we could see stars, but we could also see these clouds. And these clouds happen to be bats dispersing away from roosts in south central Texas. This is a live image. A graduate student of mine, former grad student Jason Horn, did some analysis of this. If you understand anything about Doppler radar, basically what it does is scans through a series of elevations every 15 minutes, and there's increments. You can get scans at different elevations. This is a single low elevation scan because it makes it possible to get most of the things in there. But what you're seeing here in the night sky are emanating or emerging from a series of caves and bridges and so on in Texas. That's what the night sky looks like at one elevation. With that kind of information, you can actually do vector analysis of movements of these clouds, if you want to call them clouds, of bats moving in different directions. And one of the things that Jason did, we found, and these are histograms, basically circular histograms that show the direction. And you can see the histograms are right here. These are the clouds in effect. This happens to be somewhere I can't really see it. There's a Congress Avenue bridge right here. This is what is called a Bracken Cave, which is between Austin and San Antonio. But these histograms basically show the direction of movements of these at the night. And all this yellow stuff is agriculture. All the green stuff is sort of oak savanna in this region. Here's the Edwards Plateau up here. So anyway, this kind of information has helped us understand a little bit, but there are limitations to it. It provides no information about do these bats just live on a plane, or do they have a 3D structure to their source? And a postdoc of mine, who actually is a postdoc currently, but she just got a 2-year bioinformatics grant to actually analyze the 3D structure of emerging bats. And this is that slice of, this is actually her first, she just is giving birth tomorrow, so she's been to a baby, and she's kind of laid, I guess you would expect that, right? But in any event, couldn't have been a bat. But anyway, just to give you an idea that this structure is really very dynamic. And this is, she was hoping to send me a dynamic image of this cloud in 3 dimensions, but this is the best we could do. It's a novel analysis. We're working with some folks at University of Oklahoma, who are atmospheric scientists, who've been very helpful and who do a lot of cool radar stuff. So they're helping us with this. But it provides no information on movements of individuals. That's really the important thing. We need to know that. One of the things we've tried to do is put radio transmitters on individuals to find out something about how much time they spend in flight. Bats, when we started this particular study, we put these little radios, they weigh about a half of a gram, and it turns out we can't really track them very well on land, because those of you, I think Bob Brown can probably tell you, there aren't many roads in parts of Texas, and even if there were, you wouldn't want to drive on them. So the point is, what we could do is a time budget study. We could determine something about how much time they spend. Here is a profile here. These are emergence times. The upper part of that is how much time they spend in flight. In this case, most of these bats all were out, and they came back in the middle of the night. They went out and feed again, and then they came back. And on average, they spend about 8 hours a night in flight. These are lactating females, so they come back in the middle of the night, most of them, except this one that didn't, to nurse their babies. And so this is a limitation. Again, it provides no information on distance or direction. It just gives us how much time they're spent on the flight. So one of the things we've started, and this is a colleague who was at Princeton, who's now the director of the Max Planck Institute of Ornithology in Germany. He and I have paired and teamed up to start. He's a pilot, incidentally, and I told my wife that on my bucket list is to start flying. If anybody wants to train me, I'm happy to do that, go with you. Anyway, so we actually radio tracked individual bats. I was mostly on the ground, as you can see here, and Martin Wikelski, he's the pilot, and he's also a great biologist, ornithologist, but is interested in bats, even though they're not birds. But anyway, so just a few images that we got last summer. We have 5 tracks. I'm just going to show 3 of them. What you're seeing here is this is the track. The yellow little blob is the cave. If you can follow these little guys, why it's not moving, these are supposed to be moving here. They're foraging, that's what they're staying in one place. Anyway, there was a 23-second, let me go back a minute and see if it plays again here. Somehow it might got hung up. Okay, let's try again. Okay, here it goes, here they go. Okay, they should go out and they start feeding. Okay, here we go. And they tend to, this particular bat seemed to forage or disperse and along a river. They tend to go overland, and these little dots are and eventually come back. And this was a 2.3 hour sort of route that they took. This one here is a little different, and we're again testing this refugee hypothesis about some individuals going further. These guys went out very fast one night. They were had a downwind. It turns out they were. This particular bat, we had a constant frequency transmitter on it, which basically makes it possible to measure the wing beat frequency. Okay, it'll go zzz, 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 if it's constant like this. It turns out this bat was moving at 30 miles an hour ground speed and was not flapping its wings. It was soaring down from a tailwind, and it came back at about 10 miles an hour, beating like crazy. You know, its wing beat was just going as fast as a little guy could go. So there's a lot of stuff. This is opening up some new areas that. Yes, Margaret, I'm going to learn to fly, I think. Anyway, here's another bat that, again, sort of going in the north direction, spending out some time in the agricultural areas foraging. I'll tell you a little anecdote here that just was totally amazing. That we were standing, I was explaining to some of these caves. This is a cave called Frio Cave that a lot of visitors come to every night, and I was explaining what we were doing, and I was talking to this with the little radio to the pilot and all this kind of stuff, and the guy, my pilot, Martin, was saying, well, we're right now over this farm that has 3 silos, a pond, and 2 barns, and there's a guy standing next to me who says, that sounds like my place. Sure enough, it was. It was amazing, just by coincidence that happened, that we flew over and he described exactly what was there and so on. All right. A couple other points I want to make here is that, again, energy expenditure. We know that these guys, individuals from the radio telemetry stuff, just beginning to do this. This is a piece of a grant proposal we're putting together for January, the deadline to NSF. But we're now, we also interested in how much energy they spend. Do they spend more energy or less? And this is a technique that has been developed for a number of years using stable isotopes of hydrogen and oxygen. And I'm not going to go into the details of this because I don't have time, but just believe me that you can, basically the turnover of isotopes in the body fluid makes it possible. Comparing blood samples at the beginning and after initially equilibration with body fluids, and then 24 hours later after they've done their thing, fly or do whatever, it gives us a possibility to measure metabolic rate and so on. And this just plots out the metabolic rate of free-ranging bats, both shown babies. We did this with non-flyers. The red ones are lactating females. As you can see, there's high variability there, and we would predict that high variability based on what we know now about the time budgets that we got from telemetry, and also the movement patterns and the time spent out foraging and so on. So we've more or less confirmed this theory, the refugee theory, based on those kind of data, and there's a lot more going on. Okay, some very recent sort of things with colleagues here at BU in the geography department who are using LIDAR. Is Alan here tonight, or Crystal? Oh, way up there is Crystal, okay. Well, anyway, LIDAR, again, is light detection and ranging, which this happens to be the only device available in the country and the world, actually. It's made by CSIRO, loaned to BU, Geography Department. And this looks like about the size of a rural mailbox. It scans around, and you put it on the middle of a forest, and it scans around at a certain rate, and actually gives you an image of a forest. Whoops, maybe I'm out of place here. Okay. So one of the things we want to do is use the LIDAR of an image of a forest and use thermal imaging to marry those two technologies. So we can, if the LIDAR can give us a profile of the forest, we should be able to track bats through this profile to see what effect the profile of a forest has on the biodiversity and density of activity of bats. This is actually a thermal imaged, a 3D thermal track of a bat around a light pole, and this is our simulated tree. This was our first effort to do this. So we know we can track bats around light poles. We ought to be able to track them around trees. There's the first one of the images of LIDAR to give you a sense of what the forest density looks like, the forest structure, if you wish. Thanks to Zhao Yang, who is one of the graduate students in the geography department, created this next image for me, which is a pretty cool image of LIDAR. This is in the Sierra Nevada range. This is a forest that we can ultimately, hopefully, run thermal imaged 3D trajectories of bats through to get a sense of, again, how we can evaluate forest structure and how it affects biodiversity of bats in different types of forest and so on. One other technology which I'm hoping to put into the next proposal here is the use of vertical profiling radar. Some of my colleagues in Canada, at McGill, and also some folks here in Pensacola, Florida, who track and do all of the radar work with NASA, have been developing sophisticated vertical profiling radar, which can reveal some very interesting stuff. And what this shows here is, you're not seeing double. That's a mistake right there. But anyway, this is, if you're looking up from ground level, looking up here to 2.5 kilometers above the surface of the Earth, you can individually, this radar allows you to detect individual bats and individual insects, So this actually shows, as we know, insects don't really have very strong powered flight. Most of them are lifted up by thermals during the day, and then they drop at night as the aerosphere cools. And you can see these layers of insects right in here. Up here you see birds and bats. And what's really interesting is at this time of night, all these insects down here right in this lower portion of the boundary layer next to the surface of the Earth where wind turbines are, that's where bats are being whacked. That's where they're being killed. That's where bats are feeding, where all these insects about that time of night. So anyway, this is going to provide us really some very novel new ideas and knowledge about how bats and birds use the aerosphere. So where do we go from here? Questions and research needs, there are a number of them. How do changes in the landscapes and aquascapes that we've seen affect the activities and movements of airborne fauna? How does an altered aerosphere affect the abundance and distributions of airborne fauna? How do changes in the aerosphere affect biodiversity? What impacts do invasive species have on the abundance and distribution of invasive species? And how will future changes in airborne fauna affect ecosystem and human health? And so these are questions that a few that we can begin to assess, and I'm hopeful that we'll be able to do that with support of other funding sources over the next few years. So thank you very much for listening.
Do you want to take questions first, or you want to straighten it
No,
out?
I can take questions, yeah.
Take questions first?
Yes, yeah.
So we'll take a few questions first before we go into the demo.
Okay, so I'm going to stand out here and listen to you if you have any questions.
Do mothers teach their offspring how to hunt?
Slow down. What was it?
Do mothers teach their offspring how to hunt? You mentioned that passing, and you never answered the question.
It turns out the species that we were studying with the transponders, no, they do not. In other species that we've studied, for example, fruit-eating bats in Malaysia, mothers carry their babies with them on early hunting trips. So they presumably learn either spatial positioning of their food sources relative to their roost and so on.
Hi. In the censusing you were doing in the cave, were you assuming that all bats in the cave were leaving the cave at night to forage?
Yes, and we've confirmed that by going into the cave and know that there's none left in there.
So like baby bats and things like that, they go out to forage with their parents too?
Well, it turns out that baby bats, again, are born fairly large. They weigh about 25% of their mother's weight at birth. But they're not weaned until they're almost adult size. So what's left in there, mothers, lactating females will go out to feed and leave their babies hanging in the roost. Okay. And it turns out there's a whole story about how out of a colony of, let's say, 750,000 or 1 million bats, how do mothers find their babies in that is a big challenge for mothers.
So how many baby bats per parent bat?
One. In most species, a single baby, one baby. A few species have twins, and a couple of species have 4. And the combined weight of babies at birth there is 50% of the mother's weight.
Okay. So when you guys went to the cave, you found that only baby bats were in the cave and no healthy adult bats were left in the cave at night?
Slow down.
So only baby bats are in the cave at night and no healthy adult bats? Is that
only
right?
Only the baby bats there when the mother's out to feed.
Okay.
And obviously when the mothers come back to feed their babies, they're both there.
Okay, all right, thanks.
Yeah, just to follow up on that, again, people often ask, how do mothers find their babies in a situation like this? It turns out they basically use 3 criteria: spatial memory, acoustics, and olfaction in that order. And you could do this, you can think of the same thing. Here's a room, and you're a mother bat, and you leave your baby over in this corner, and you'll know, you could close your eyes and you can know where you were in this, you could find that corner of the room just by spatial memory. You do this at night, you have to go up into the bathroom, you walk around like this, and your eyes are closed, and you find the bathroom, okay, by spatial memory. You know how many steps you took to get there. It turns out that spatial memory is the first cue. Then the mothers land on the bats, they crawl around, and babies are crying, "Mommy, Mommy, I'm hungry," and mommies call back to them, and eventually they get very close, but the final decision is based on how they smell. And mothers do recognize their own babies by odor. Babies don't recognize their own mothers. Babies are very aggressive. They'll grab on anything, even my finger, to go try to find milk. Okay, I thought you might be interested to know a little bit more about thermal imaging here, and I put this up here just to show you that this works independent of day or night, and you can see a lot of different features here, some of which I don't necessarily want to show, but you do see hot spots, okay? Now, I'm going to I'm going to take my shirt off, my coat off, and you can see it's quite different. Okay, I'm a little sweaty right now, probably, but I'm also warm here because my shirt is close to my skin. And this is something that actually this technology is actually used in medicine today. Instead of having mammograms, it turns out that these high-end thermal cameras are actually used to detect breast cancer. So there's other kinds of many uses of thermal cameras other than just playing them with them like we're doing right now. But the point is, so I'm going to ask Nate Fuller is a grad student of mine. He's going to scan the audience here, and you can actually look at yourself here as you wish and see what hotspots you might have that you don't want to show. You can all stand up and raise your arms. You can see your hot armpits probably as you go through here. You may not want to stand up to see your other hotspots. Actually, I do this in my class sometimes. When I do it in my ecology class, I say, okay, everybody stand up, and they'll go, oh no. But anyway, it's something that we have a lot of fun with. Actually, well, I won't go any further on that. Okay. So now, what I'd like to do, and you notice I had, do I have a redneck on my back here? Get back at me. Do I have a redneck? I'm going to test a hypothesis here. Okay. Do I have a redneck? Okay. I'm going to test a hypothesis because our president is from Texas, and you know that is the source of redneck country. Okay. So Bob, you're going to have to come up here, and we're going to look at you and see where your hotspots are, and see if in fact you are a hothead or a redneck, one or the other. Okay. You got to stand about where I am here. Okay. Where are you? Okay, so pretty red. Pretty red. Okay. All right, and we can't let David off with this too. I mean, you know, provosts have to see whether they have hotspots too, and he probably has. I'll get out of the way. Oh, it's red. Yeah, you got. But anyway, but yellow, you know, the yellow shows here because we have a little fat in our cheeks. Okay. Okay. No, seriously. No, okay. So anyway, anybody else have a question? I just wanted to have that with Nathan here was doing this. Anybody have questions?
Just don't turn this over to the TSA. We haven't had any questions about ecological impact of some of this stuff, the white nose disease or the ecological impact of the decrease in bat populations. Can you say something about that?
Sure. Well, I mean, one of the things that, you know, anytime you put a talk together like this, you end up starting with about 100 slides and then you throw out 20. And I felt I wanted to talk a little bit about the thermal stuff, and we didn't talk about white nose syndrome. But as you know, and as David said at the beginning, actually white nose syndrome, which is a fungal disease of hibernating bats for the most part, started in the Northeast, has had a devastating effect on bat populations. Those of you who live out in the country or may go to the country periodically, you just don't see bats very much anymore. And this fungus affects, invades the skin through the sebaceous glands and the hair follicles, does seem to cause irritation to them. The bats tend to arouse more frequently as a consequence of this. They basically run out of fat. Hibernating bats put on fat in the fall of the year, but and normally they survive throughout the winter by utilizing that fat. Bats that have white nose syndrome run out of fat in midwinter, and they start sometimes going out flying in the snow and so on, and damages their wings, their frostbite, they get scarred tissues from the fungus and so on. And right now we don't know what the cause is. There's a particular hypothesis right now that this is an invasive species, the fungus. That is, there's a species of fungus that is, actually genetically it's been shown to be almost identical to a fungus that comes from Europe. It was first introduced to New York State near Albany in Howell's Cavern, which is a commercial cave, and in the winter of 2006. Since then, it has spread at a rate of about 200 miles a year. It's now in 9 states, and it actually is in West Virginia and Virginia. Pennsylvania and West Virginia. And it's very close to some other major hibernating colonies that then disperse out in the landscapes during the summer to feed on insects that feed on crops and feed on forestry and so on. So the impact here, there's a really serious potential economic impact of these losses of bats. We've estimated there's probably 1 million bats that were lost in the last 3 years here in the Northeast. How many more have been killed or are dying now? We don't know. We'll know a little bit more this winter how many. Some of the hibernating colonies have declined by 95%. And these bats eat a lot of insects, okay? Often they eat up to their body weight a night in insects during times of the year when the energy demands are highest. On average, probably they're half their body weight a night in insects. And I did some back of the envelope calculations based on the loss of a million bats in the last 3 years. These bats would have eaten the equivalent of something 694 tons of insects that were now not being eaten. And this is just the tip of the iceberg. We don't know how to stop it. Our lab is working on the immune system. Our lab is working on the body composition of these bats, trying to understand just what's going on. We're doing other work, and we have proposals out to do more. I'll tell you one of the more sad stories here, I think, and that is that I actually testified in Congress to try to raise money for this. I was invited along with several other people, including the regional director of the Fish and Wildlife Service. I worked with about 20 scientists to put together a budget, a 5-year budget for how we could address this question. We came up with a budget we thought was a very reasonable one. It was actually over 5 years, $55 million. Now, it sounds like a lot of money, and it is. But the congressional folks, the House committee a joint committee in the House, actually heard our testimony. And they didn't recommend any money, none. The Senate committee recommended $500,000. Now, it's not a done deal yet. We have some very, very supportive congressional folks who are working with us. We're trying to get another $3 million here and there, but probably our best bet right now is not through the Department of Interior, that was the money because our Fish and Wildlife Service folks are, in fact, our stewards of our wildlife. And so the money would go into the Department of Interior. But there are other sources of money. NIH, in fact, NSF, even though this is not a human disease, it's possibly linked to, it could be linked to it. And I just came back from a meeting at NIH 2 weeks ago, and they're very interested in bats and emergent diseases of bats. And in fact, I've been talking to Mark Klempner and his gang about this, because I think there are a lot of things, there are a lot of the canaries in the mine that we don't really know about right now. And I think we have to be alert to this when we get losing big populations like we're losing now, and in the future, we have to be ahead of the game. And I think our lab is, at the leading edge of those studies. So I don't know if I've gone overboard, but if you have...
Not in my opinion. Are there other questions? Yeah.
Yes.
John.
How aware is the Agricultural Committee about the danger posed by loss of bats? And then a corollary question, are there any funds possibly forthcoming from agricultural organizations to help your project?
Well, Department of Agriculture is aware of it. And certainly that's another possible source of money. The problem is right now we're in difficult times in terms of economic issues regarding federal government. Healthcare is at the top of the heap, and everything else gets shoved down, in the bottom of the pile. So they're certainly aware of it, and they're concerned about it, but not to the point where the checks are written and handed out to us. But we're working on it. And I have to say, students in my lab, and undergrads and grads and postdocs and so on, are all helping with this, apart from their normal kinds of things that they are doing in terms of their thesis work or whatever. We sort of didn't put that stuff on hold, but we're doing more things. And so this is kind of our night job, in effect, because we feel that's very important to work on. So if anybody, yes? Well, there are some parallels to the colony collapse in bees. You can name a number of problems that are happening to our wildlife. Bees being one, colony collapse, the chytrid fungus killing off frogs around the world. These are all different causes, but in effect, collectively, they are canaries in the mine telling us that our environment has problems that we need to really address, and it takes teams of people to really address these questions. But yeah, there are parallels, but no actual direct links between any of these that we know about. Okay, anybody else? Okay, yes.
On average, how long are the bats out of the cave at night altogether?
On average, depends on the species. The freetail bats that I pointed here during lactation, they're out 8 hours a night. interesting little anecdote about this I must tell. Several years ago, one of our undergraduate students did a study. Well, we go back a minute. One of my graduate students actually ended up getting sick, and we had to take him to a hospital and do blood and so on. And we thought they can analyze blood for cholesterol and all kinds of things. And so we took some bat blood in there and we analyzed it. Now, because we were interested in whether these guys might have high cholesterol levels, because half of their diet is fat. If half of your diet was fat, your arteries would be clogged, right? These bats, the arteries in their heart were clean as a whistle. What's the answer to that? You fly 8 hours a night, you'll have clean... So, exercise. Keep those arteries clean. Okay. Anybody else? Okay, now before we leave here, I have actually something. Oh, yes.
Well, we have something to give to you, but please go ahead.
No, you do the...
Oh, no, after you.
Okay, all right. I'm going to put this in my pocket. Well, just so you know that I'm
Hard carrying.
that's right. I mean, my day job is to teach and to mentor and answer telephone calls and all this kind of stuff. But my night job, as my wife would tell you, is sitting at the computer and writing, and that's what it is most academics. We don't have time during the day to do much other than see students and answer phones and do email and so on. Well, anyway, for the last several years, I've been working on another book, and actually David mentioned this. This is a book that just came out Friday, and I have a copy. I haven't signed them yet, but Bob, if you come up here to accept one, and David for the other, you can use it as a paperweight if you wish, but I hope you will glean through it periodically to find out what bat biologists actually do.
You haven't signed it yet?
I haven't yet, but I will shortly. I'll do it. Yeah, okay. You're welcome. Okay. Okay. Okay.
So, apart from what you learned about bats tonight, and you learned a lot, it's all going to be on the quiz, what you really learned is the passion that our faculty have for the subject, the deep passion, and how it extends, and how it reaches out essentially to all knowledge, how it's all linked. I think it was an absolutely fabulous presentation. Let's thank Tom again for a really wonderful talk. And in recognition of the presentation tonight, we're giving him a copy of the framed poster. Tom?
Thank you very much.
And something which in this economic climate he might appreciate perhaps even more, token of our gratitude. Great job.